My name is Abdulkader Rahmo. I studied actually biochemistry, but I spent my last 12 years working on a unique type of stem cell that should allow us overcome a lot of hurdles and bottlenecks that plagues cell therapy and specifically regenerative cell therapy. Because of that, I built a company which is SMSbiotech about 10 years ago, and it is located in San Diego County. So why stem cells and why cells? Cells, it's because the smartest drug is a cell, and a stem cell is even superior. It's the smartest among other cells. And we think that true regeneration can really occur through stem cells, mainly because of that inherent intelligence, evolutionary based, not necessarily AI based. What are the bottlenecks regarding cell therapy and specifically stem cell therapy? There are many. Safety, safety, potency, scalability, targeting, engraftment, and survival. That's why we have very little success with regenerative therapy today. Well, with SMS cells, we overcame all these problems because the cells are isolated from adult human. These are adult stem cells isolated from peripheral blood. They include no genetic manipulation. They don't need any genetic manipulation. The cells have the ability to multi-target several cells and to orchestrate a process that is quite complex, that requires the regeneration and rebuilding of tissue. Destroying a tissue using a CAR T, for example, is easy, but rebuilding a tissue is a very complex matter. These cells can be cell biomanufactured without almost limitations. So we can grow them for more than 27 passages without loss of potency. This is unheard of for adult stem cells. They are strongly selective in that they actually directly bind to specific cells. That means you can actually see in this case a stem cell binding to a specific target cell and measure its potency therefore easily. They can be administered directly to the lung non-invasively using a nebulizer because they are so stable and small enough to cross a mesh nebulizer and be administered directly without any damage. They are really resilient towards different conditions, and they meant to be this way because they are the emergency cell. They are present in the peripheral blood. Any cell that is present in the peripheral blood has huge values. It circulates. It does vital issues to the body. And in this case, we think that SMS cells target regions where they require regeneration. So, these SMS cells have a diameter of about 4 micrometer, quite small. They are very transparent and very fast in motility. They actually move visibly. You can see them visibly moving. You don't need a recording for that, which is a very good reason for a cell biologist to throw them away because he will mistake them as a bacteria. And they escape, therefore, detection by their cell biologist for a long time until we discovered them. I will present here a video with the help of our friends over there. In this case, you will see how the cells will escape the glass capillary, the SMS cells tagged with the fluorescence signal. They will escape the glass capillary and they will go and bind to mesenchymal stem cells in cell culture. They bind to them strongly and they stimulate their proliferation. Now, knowing, is the video working? Nope. Move back. No. As I said, they are tiny cells, hard to find. So the cells will bind to the mesenchymal stem cells, and they will continue moving with the mesenchymal stem cells, and the mesenchymal stem cells will happily divide and grow and migrate. And they actually stimulate the proliferation of mesenchymal stem cells in ideal conditions. Even if you put the mesenchymal stem cells in ideal condition, you still get more proliferation by introducing SMS cells. Now, this is very important because mesenchymal stem cells are almost present in every vascularized tissue. There are few that are not vascularized. Any vascularized tissue contains mesenchymal stem cells. So this is a basis for a platform technology. Now it's working. Yeah. Okay. So you see here the cells in green escaping and binding now quickly to the mesenchymal stem cells. The mesenchymal stem cells moving and carrying with them the SMS cells. Now, this binding is so strong that when you detach the cells, both cells are detached together. They still link to each other. Now, in biology, we know if there is a natural strong binding, this is not done for nothing. There is important alignment here between the two types of cells. And you can see them carrying the SMS cells, like here, for example, and another one here. Yes, and all over. So this is the basis for an endogenous cell therapy. So the cell therapy is not aimed at having the SMS cells create a new tissue. It's actually affecting endogenous cells and creating the tissue based on the host cells. Therefore, the end product, the end tissue product, is completely compatible with the body of the host or the patient. So it's an endogenous cell therapy. This is, of course, a patented regenerative platform technology. We received so far 6 patents worldwide, and we filed for about 15. Now, it is, of course, not just binding. Binding, for example, to another key cell in the lung is binding to alveolar type 2 cells. Alveolar type 2 cells are strategically important in air sacs because they are progenitor cells. We know already that they are responsible for the repair and maintenance of air sacs. They bind strongly to alveolar type 2 cells. They do not bind to alveolar type 1 cells, which are not progenitor cells. But progenitor cells, like alveolar type 2 cells, can produce alveolar type 1 cells. And when they bind the SMS cells, when they bind to alveolar type 2 cells, there is a lot of gene expression changes that occur. And these involve central aspects of overall healing, including countering inflammation, countering fibrosis, countering apoptosis, countering blood vessel permeation, countering vasoconstriction, countering pathogens, pro-angiogenesis, pro-parenchymal growth, pro-surfactant production, and even pro-DNA repair. Now look how many pathways, how many genes are involved in the countering inflammation. So targeting inflammation, definitely a good subject. And when we look into these gene expression changes that are based on natural cells, what we're doing here, we are really decoding regeneration. We are understanding what pathways are necessary for regenerating the tissue, because we're talking about natural phenomena, natural mechanism, natural cells. Another aspect of these cells, which is kind of amazing, is their ability to produce huge amounts of extracellular matrix, which is kind of surprising. A tiny cell producing such large amount of extracellular matrix? Yes, indeed. The extracellular matrix that is produced by these cells is able to actually differentiate endothelial cells into small tubuli and create large vessels, as we see here in this video. It's a de novo creation of a large vessel affected by the extracellular matrix of SMS cells. The large vessel that is formed is visible to the naked eye. This is how strong angiogenic you need a cell to be to exert regeneration. Of course, there are many other proteins that we can learn from that are produced by the extracellular matrix, by the SMS cells, extracellular matrix, that can be identified and understood and studied. One of them is, for example, heat shock protein 90, the secreted form that is important for repair of protein structure. We tested the cells for the regeneration of air sacs, and we created a model for lung emphysema in rats by introducing the enzyme elastase. So elastase will destroy the air sacs in a short period of time, and we introduce SMS cells, single injection, after a week. We waited for 1 week and we analyzed then the structure of the lung of the rats. And what we observed is that in just 1 week, we had 60% regeneration of the air sacs, structural regeneration and functional regeneration, because 60% was enough to make these rats run like a normal control, healthy control, not affected by elastase. But when we used another injection of SMS cells, we had a dose effect, and we got 100% restructuring of air sacs in less than 10 days. Such a strong effect cannot be explained unless we understand their unique mode of mechanism. We are starting a phase 1 trial in Australia very soon, using 18 COPD patients, mild to moderate, and that will be using 3 levels of the SMS cells and a cohort, 3 cohorts. Now, the exposure will be using actually a nebulizer, because the cells, as I said, are resilient enough so that you can nebulize them without affecting their viability, their ability to proliferate, and their ability to bind. They will engraft easily, which was a huge problem for stem cells, because they bind to a specific target of cells. The proliferation of alveolar type 2 cells will ensure also the regeneration of alveolar type 1. We will have gene expression. We will have gene expression changes that are important for the overall health, and I described some of them in the past slide. So we will also stimulate the proliferation of the endogenous lung mesenchymal stem cells, and ECM regeneration will ensure the restoration of the air sac wall. We will also generate blood capillaries, because without those, there will be no functioning air sac. But there are other indications that can be used from these cells, obviously. One of them, an early one, was to use the extracellular matrix for the wound healing of diabetic mice. And for that, we used splints to prevent actually the contraction of the skin of the mice. And we tested this system that is closer to the human injury or wound, and we saw a 250% average increase in granulation and 120% increase in epithelialization, and also the strong formation of blood vessels. We also tested the cells for joint disease, and indeed we found recently that SMS cells interact also with chondrocytes and bind to them, and that in addition to the fact that they bind to fibroblasts is quite important, in addition also, of course, to the secretion of extracellular matrix. So we had very good results with dogs, extraordinary results with companion animals. We did also an example of intravenous systemic injection of the cells into a dog that is 11 years old. He belongs to one of our investors. So he wanted a proof of concept. We said, okay. So we injected that dog that the owner thought is gonna die before we get to commercialization. He knows about the FDA. So, and what happened is, after the second dose, 3 days after the second dose, the dog that was always laying down and not moving, quite lethargic, this is what happened. He's playing frisbee. So from that, we conclude that the cells actually can have the ability to home in to the right location, because we don't know exactly what's wrong with that dog. They know more about that. So we intend also to apply the cells for other indications, especially for pulmonary because of this ability to administer the cells directly to the lung. But we're looking also to other organs, especially wounds and joint diseases, as I mentioned, but also cosmeceuticals. So SMS cell therapy prospects for tissue regeneration are unique because they target several types of cells key to tissue regeneration overall, and that are present in many tissues. So we're talking about a true platform here. There is a reason why the cells are systemically present in the circulation. SMS cells stimulate proliferation of parenchymal cells, such as the alveolar type 2 cells that are key to lung regeneration, which is an excellent target. SMS cells activate mesenchymal stem cells that are endogenous promoters of tissue repair. There is enough research in that field, I think, is more than enough. And SMS cells inhibit actually fibroblast cell proliferation, which is the main cause of organ fibrosis. So we think that there may be a way to inhibit fibrosis in multiple organs using these cells. Unlike mesenchymal stem cells, SMS cells proliferate extensively without loss of potency, ex vivo, and they are sturdier, more robust, easier to handle and store and administer. You can store these cells in suspension at refrigerator temperature for 3 weeks. This is unheard of for cells. SMS cells can be administered using a nebulizer and can be introduced directly to the lung. They are unlikely to induce any significant immune reactions because they have, like other stem cells, very low major histocompatibility proteins. These are adult stem cells, and therefore they are safer alternative than using embryonic or genetically engineered cells. The bloodstream is one of SMS cells' natural habitat. And given the size, they can cross any capillaries, unlike, for example, cells like mesenchymal stem cells that get clogged in the capillaries and therefore die in quick numbers. SMS cells demonstrate in vivo, using animal model, an extraordinary safety and efficacy profile. I didn't speak about the safety profile. We tried so much to produce any adverse reaction using these cells. We used 20 times per gram what we're going to use in a human and still didn't detect any adverse reactions in the animals. I would like to thank our team for this excellent support. And I would like also to mention some of our advisors, Professor Peter Wagner, who is a COPD expert, world expert, Professor Ghassan Kassab, cardiovascular expert, and Professor James Shirley. He came from the MIT. He's an adult stem cell expert. And Dr. Jason Kirkness, he's still a chairman at the American Thoracic Society and works for us very closely, and Professor Gary Seek from the Mayo Clinic. But important is the message, not the people actually here. The message here is we have SMS cells availability, it's accessible to common diseases, and stability, manufacturing, administration, logistics, binding, targeting, engraftment, effect, molecular, cellular, and organ level, and safety, doses range, right? So all demonstrated through preclinical studies and soon via clinical studies. Thank you very much.
Thank you. We have time for maybe a couple of questions. The usual.
It's really amazing talk, and I think it chimes very well with previous talk. The concept is very old. You reduce inflammation, you reduce regeneration. So would it be correct to say that those cells bind to MSCs exclusively?
No, they bind to multiple cells because you see, multiple targeting is very important. The tissue rebuilding, it never hinges on one cell. You need multiple cells, multiple pathways, even within the cells, to exert really a complex construct, like, for example, the air sacs. It's like imagining building a home. You need many experts, leaning on that task. You need one expert to demolish it, but you need multiple experts to actually build all these complex components. Mesenchymal stem cells are important, but they are not enough by themselves.
So do those cells carry a nucleus?
Yes. They carry a nucleus, otherwise they wouldn't be able to proliferate, right?
And when you inject those cells into animals, are these cells autologous or non-autologous?
Oh, that's a good question. Our treatment is allogenic, off-the-shelf treatment, not autologous. That's what we're aiming for. That's the gold standard that everybody is aiming for. And in all our experiments with the animals, we used only human cells, never animal cells.
So that means that their survival systemically is very short.
There is?
They survive very short time.
Oh, yeah. We checked on their viability inside the body. It depends on which tissue you inject them. In the lung, their presence is about 2 to 3 weeks. Yeah, but the binding occurs within hours. Gene expression changes occurs within 3 days. So it's enough time to do and exert the effect.
And using SMS cells for treating Alzheimer's disease or Parkinson's to regenerate
neuroregeneration we did not test. We shied away a little bit out of that because if we get into that, the FDA will be watching everything we do very closely. It may affect the brain. But definitely it could be a target. Now, I need first to understand the mechanism. There may be possibility of interaction between SMS cells and the glia cells. And we're looking into that. We have a very good partner, which is the military. And we're looking with them into many aspects of other applications.
Okay, thank you very much.